Okay, well, it's a great honor to be here. It's actually a terrific pleasure to see so many people from Africa in the room. I don't very often get to speak to a lot of Africans. Is this clock going to get started so that I will know where I am? Someone can do this? Anyway, what I'm going to talk to you about today is I'll give you a little bit of biography to give you some idea of why I got asked to come and talk to you. But I'm going to focus on bacteria. And I'm going to focus on bacteria for a good reason, and that is that finally scientists around the world, but especially policymakers in the US, have actually figured out that one of the reasons that we work well as humans is because we carry an awful lot of bacteria around with us. And if we kill all of the bacteria that live with us, we will die. And that's probably not such a good thing. Now, I first got interested in bacteria when I was at Harvard. I did a postdoc at Harvard. I was educated in England in organic chemistry, Sheffield University. Went to Harvard and started working on transfer RNAs that were involved in bacterial cell wall biosynthesis. And then moved on to Cold Spring Harbor, and I'll tell you about some of the things that I did there. And most recently have moved to New England Biolabs, a company in New England, just north of Boston, where we make reagents for research. And the company is actually a rather interesting one, because not only do we try to promote research, but we also try to promote science in the third world. And many years ago, we made a decision that we were going to start working on diseases, tropical diseases. We work on filariasis. In fact, since we got started back in the 1980s on that, we have spent more money on filariasis than the National Institutes of Health. And we did it only because we were interested in trying to help to solve this disease, not for any commercial reasons whatsoever. Now, after I finished my postdoc work, I went down to Cold Spring Harbor Laboratory, and I was there for a total of 20 years, from 72 to 92. And when I first got down there, I was asked to sequence SV40 DNA because Jim Watson had just managed to get a big grant to study cancer. It was at the time when Nixon had declared a war on cancer, and there was just huge amounts of money floating around. There was way more money floating around than it was possible to spend in any sensible way. And so a number of us benefited from that and were able to do things that otherwise might not have been done. So I was hired to sequence SV40 DNA. When I got there, I discovered there were two other groups already doing it. It didn't make any sense for me to get started on it. And it was at a time when the very first restriction enzymes had just been discovered by Hamilton Smith, by Werner Aber, and it seemed to me that these enzymes actually offered a new possibility for sequencing DNA. At the time, the only way you could sequence DNA was by copying it into RNA and then sequencing the RNA. So what happened was when I got there, I forgot about SV40 and started working on restriction enzymes. I made the ones that were known, and then we started to look for more. And of course, everywhere we looked, we found them because something like 90% of all bacteria have restriction enzymes. These are the enzymes that protect them against bacteriophage infection. Now, by 1975, we had 30 enzymes that had been discovered. They were not commercially available. You could get them if you came to my lab. We sent them out to people. People would show up at meetings with DNA, and we would let them go and take a dip into each of the different enzymes to see if there was one that would cut their DNA. And it was clear that these were going to be very useful. In fact, if you look back now and think about the biotechnology revolution, without restriction enzymes, it would not have happened when it did. And so these things really that were not believed to be of any great interest to start off with actually were one of the workhorses of the biotech revolution. Now, I tried to persuade Jim Watson that we should start a company at New England Biolabs to sell these enzymes, at Cold Spring Harbor to sell these enzymes. He, A, wasn't interested, B, didn't think you could make money at it, and C, thought it was rather dirty to be connected with industry. Fortunately, I ran into a fellow called Don Combe, who had started New England Biolabs, who had also had an idea that perhaps he could sell these enzymes. And so I went and I became their chief scientific advisor in '75. When I say they, at the time, it was Don Combe, his wife, and one technician. And between us, we made $200,000 selling these enzymes the first year of operation. So we didn't need a venture capitalist. We didn't need anyone to invest. We made money the old-fashioned way, and we earned it by selling products. Also in 1975, I produced the first list of all the known restriction enzymes, which became the precursor of Rebase, which is a database I run. In '76 onwards, we started to clone the genes for restriction enzymes, and then a little later, we started using bioinformatic methods to study them and to see what was going on. And this was really the start of bioinformatics, too. Now, in 1977, I took a slight diversion. We were looking for some interesting questions to ask that we could use the restriction enzymes to solve. We became interested in transcription. Everybody else at the lab was working on this virus, adenovirus, and we thought it would be fun to try to characterize a promoter in adenovirus and see if we could work out whether promoters in higher organisms were the same as those in bacteria. Well, of course, it turned out they weren't. Things were different. But along the way, completely by chance, we discovered RNA splicing, and that was the work that ultimately led to the Nobel Prize in 1993. But it was a sideline. It was not my major interest at the time. Now, later on, after we'd been cloning a lot of the restriction enzymes and a lot of the restriction enzyme genes, when you clone them, you also have to clone the methylase genes that come along with them that protect the DNA against the restriction enzymes. And we were interested in doing a crystal structure for one of these enzymes. And in the process, we discovered a phenomenon called base flipping. And that comes about because when the methylase binds to DNA, so normally you've got a nice double helix of DNA, what the enzyme does is to flip the base that it's going to methylate 180 degrees right out of the helix. And that you can see just here. This yellow is the cytosine base that is going to be methylated. And this was the first time that this phenomenon had been discovered. And sort of like splicing, it was a nice one. No one had predicted it. And it's always fun to make discoveries that no one has previously predicted should be there. We immediately speculated that perhaps there were other enzymes that would do chemistry on DNA that would behave the same way. They would flip the bases out of DNA. Because in fact, if you look at a typical helix coming down here, the bases are stuck in the middle of the helix, and it's not easy to get at them. It's not easy for an enzyme to get in there and actually do some chemistry on them. And we now know that every enzyme that has been discovered so far that does chemistry on DNA uses exactly this mechanism. It flips the base out of DNA in order to get at the base and do the chemistry. There's one other interesting feature of this, and that is if you look at this helix, what's happened as soon as this base has gone, you've actually disrupted the pairing interactions, the stacking interactions between the adjacent bases. And you can think if you wanted to then flip this base out of DNA, it would be quite easy because half of the energy holding it in place is gone. And at the time we speculated, well, perhaps this was the way in which you unwound a helix if you were going to replicate it or transcribe it. Well, it may surprise you to know that between 1993 and now, we still do not know how the first base is opened up by either an RNA polymerase or a DNA polymerase. But interestingly, we've recently come across a case of a methylase that's not a particularly good methylase, but looks as though it's involved in replication, in plasmid replication. And we're currently doing experiments to see if, in fact, this is the way in which this particular enzyme opens up the helix. And what's happened is that it's actually become a helicase. It's become a way of opening up the helix for replication. Now, since I went to New England Biolabs in 1992. The first thing that happened, of course, was I won the Nobel Prize in 1993, which was very nice, something I would recommend to all of you. And I certainly hope that along the way, some of you will emulate that. You know, there are some very good Africans in the past who have won the Nobel Prize. But one of the things that I've been doing since I got there is cloning restriction enzyme genes. We wanted to make sure that we could make all of the enzymes from clones rather than from the original organisms. And that meant cloning something like 250 genes. For the first couple of hundred or so, we could do it by using what I would call fairly traditional methods. But later, we decided it would be simpler just to sequence the genomes of all of the organisms that had them in, and then find the genes and then go on and do it that way. And basically to use bioinformatics, to use our ability to look at a DNA sequence and predict the genes that were there and move on from there. And that led to another project which I call Combrex, which is designed to try to make predictions about what genes are doing and then to test those predictions biochemically. And this is really important because it is going to be the foundation for biology in the future. If you look at what's happening, people are sequencing everything they can think of, but no one is doing the biochemistry or the genetics or the basic biology to understand how these organisms work, and we have to do a better job of predicting function from DNA sequence. Now, there are a couple of things that we've been doing recently that I'm very excited about that relate to bacteria. One of those is bacterial epigenetics, and we know that in bacteria there are many DNA methylase genes that are present. There are all of those associated with restriction enzymes, but there are others too. But until a couple of 2 or 3 years ago, it was not possible to actually look and see what was going on there. It was very difficult to work out methylase recognition sequences. I also want to point out that when we look at a typical human and we look at the bacteria that live with us, there are 10 times as many bacterial cells as there are human cells, many more bacteriophages, and we don't know how many. And one of the ways in which bacteria protect against bacteriophage infection is by using restriction enzymes, among other things, to stop the phages from getting in. And so methylation is something that the phages have figured out. If they can methylate their DNA ahead of time, they can overcome restriction barriers, and we find quite a lot of phages do that. But we also know that there are ways in which bacteria methylate their DNA to regulate genes and to do various things. So I've got a little list here of why bacterial DNA methyltransferases are interesting. They're part of restriction modification systems. They're easily found by bioinformatics. Some of them have regulatory activity. They support epigenetic phenomena. They control gene expression. They're very good examples of proteins that recognize specific DNA sequences. And the one I'm most interested in is the one at the end, because what we're finding is that there are many methylases that are in bacteria that have absolutely nothing to do with restriction modification. And so the question is, what are they doing? And some of these are incredibly well conserved. Some of them maybe only methylate a half of their recognition sequences or a third. But my gut feeling as a researcher is that there is a big discovery to make here. And to be honest, I have to tell you, I love making discoveries, but in this case, maybe it would be okay if one of you made the discovery, because there is something big going on, I will guarantee it, but no one has any idea of what it is at the moment. Now, I just want to tell you a little bit about PacBio sequencing. This is this method that allows us to get at methylase genes very easily. It involves basically copying a single molecule, an unmodified molecule. You don't do any cloning or anything first. And when the polymerase comes across a base that is partially methylated or fully methylated, it slows down. It takes time to add the base opposite a methylated base. And because the method measures the time it takes to put a base, a new base into DNA, you can see where the methylation has taken place. You can then look and see, well, if I look at the surrounding sequences, is there a common sequence that it sits in? The answer is usually yes. And in this way, we now have data for many hundreds, if not thousands, of new DNA methylases and their recognition sequences. And to give you some idea of the dramatic effect this has had, if we just look at so-called type 1 and type 3 methyltransferases, these are ones that are part of restriction systems that are among the most effective at stopping bacteriophages from getting around. From 1970, when the very first of these methylases was discovered, up until 2012, there were maybe 40, 50 of these that had been studied, where we felt we had a good idea of what their recognition sequences were. And in the last 3 years, we're up above 1,000. The rate of discovery is going up and up. The variability in these sequences is enormous, so if we find a new one in a new genome tomorrow, the odds are very high it's going to be a new specificity that no one has seen before. So there is a lot of methylation going on in bacteria, and it is not well understood. Now I want to switch gears a bit and tell you why you should love bacteria. And I use this in the context of probiotics. Okay, bacteria have gotten a really bad rap because for many years the microbiologists, the only thing they were interested in were all the pathogens. Right? And so we heard all about pathogens, and everybody thought bacteria were bad. Well, in fact, bacteria are good for you. Something like 99.9% of all the bacteria living with you actually are good for you. They're not trying to kill you. They're not doing anything nasty. They're good. This, of course, is a good source of probiotics. Lactobacillus and bifidobacterium, which you find in yogurt, are very good. But I want now to talk about one instance that I find particularly compelling. If you go to a hospital in the West, if you spend any length of time there, there is a very good chance you will get an infection with Clostridium difficile, C. diff, people call it, which is increasingly resistant to all the antibiotics we have. Now, C. diff doesn't kill you, usually, but what it does is it produces a diarrhea that is intermittent. It comes unexpectedly, and so people are sent home, and usually they never leave their homes again because they don't know when they're going to get an attack of diarrhea, and they don't want to be out if it happens. And it makes life really miserable. Now, in the US, they try to treat it with vancomycin and fidaxomicin, but if that doesn't work, there is only one method left, and that is something that's called a fecal transplant. So it turns out that if you make a nice slurry of feces and capture the bacteria and introduce the bacteria into patients, assuming this has come from a good, healthy fecal transplant patient, then that will cure C. difficile 95% of the time. There is something in the normal gut flora that stops C. difficile from growing. At the moment, we don't know what it is, but we know that it works really well. This is something that is being practiced in hospitals in the US. It's also being practiced in hospitals in the UK and in some places in Europe. Regulation of this is a little bit iffy at the moment. The insurance companies in the US are very worried that if something goes wrong during a fecal transplant, there will be a problem. But I love these next couple of slides because they also give you some indication of problems in the US. So Libby Homan at Mass General Hospital lent me these slides. And so, of course, you have to ask, well, how do you get the feces into a patient? And so she developed some gels, so you can isolate the feces, put it into a gel. Unfortunately, the first ones were translucent, which didn't do anything for the patients. No, that's a joke. It's a joke. Okay. In fact, you do not see what has gone into these pills. You take a couple of these, and usually one treatment is enough. Occasionally, you need a second treatment. Of course, one of the advantages is you minimize the risk of a bad aftertaste if you do it via a pill. And now, UPS, Universal Parcel Systems, they have this advertisement, what can brown do for you? Well, in the case if you have a C. difficile infection, brown can do a lot for you. But of course you have to worry wonder about what else. What else is there in the normal gut microflora that might be helping you? So this is just a list of why bacteria get such a lot of bad publicity, why people don't like them. These are all the nasty diseases that they produce. But in fact, I think we have to think of bacteria in a totally different way. So what's happened? Bacteria have made us their home. And when you buy a home, what's the first thing you do? Well, you surround it with a fence. You put a nice fence, maybe a moat up there. Maybe you put in an alarm system so that if something bad is going to happen, you keep off the invaders. And this is what bacteria do for us. Bacteria have no interest in our getting sick. They're only interested in protecting us. And we know that they produce compounds that will kill other bacteria. They will kill pathogens. We also know that they're capable of taking care of cancer cells. They produce anti-cancer agents. And I think the bottom line is, as we find out more about this, and if we can work out how all of this works, we really have the opportunity to both manipulate our current microbiomes, to make sure that our microbiomes are healthy. And the odds are we can do all of this without worrying about antibiotics, without getting involved in that. And I think we're going to be able to lower the costs of healthcare to a point where in the developing world, this will become a very serious attribute, a very serious help for healthcare, and in the West too. So I thank you all for your attention.